Yes, good afternoon. And first of all, I would like to thank Haldo Stephenson and 31st Connect and the organizers for the invitation. I'm here for the University of Hamburg, where I'm involved in technology assessment of pharmacogenetics, and I will come back to that a bit later. And I'm working for the Sunshine Project. That's a non-governmental organization in Germany and in the USA. And we are basically working to strengthen the norm against biological weapons. And that's why I'm here. I think it's commonplace today that in history, every major new technology has been used for military purposes. And I think what we are now facing is the threat that the biomedical sciences will also be abused for military purposes. And I think the consequences might be devastating. It could affect our lives, not only future ways of warfare, but our lives, because biology goes so deep. It's our life, our food, our environment, our brains, our behavior, everything is in a way connected to biology. And if we allow a hostile use of this biology in the future, it may go much, much deeper than any previous weapons development. And what I basically want to do in this next 20 or 30 minutes is to raise three issues. The first one is we are not any longer talking only about infectious diseases. A hostile exploitation of the biomedical sciences lurks everywhere, and I will give you some examples for that. The second issue is we are not only talking here about terrorist groups or so-called rogue states. I think we have to have a very close eye on our own governments, because the new technologies trigger new desires also in our Western countries. And then the third question is, what can we do about it? I will try to raise some ideas for solutions for these problems. My introductory slide has been stolen now by Volker Beck, because everything that's on here has been addressed by him already, so I go through that. That's antibiotic resistance and plague. That's the invisible anthrax, and both of which have been done in the former Soviet Union program, and some general ideas of what could be done using genetic engineering or genomics. And he also mentioned the poliovirus. I don't have to say anything about that. The only thing he left over for me is the UV resistance. One of the major problems these biowarriors have is that their viruses and bacteria are unstable in bright sunlight. DNA is rapidly destroyed by the UV light. But now there are ways to get around that. There was one experiment, not using an infectious agent, but just Escherichia coli, to introduce suntan genes, I call them, carotenoids, which protect the DNA from UV damage. So there are abundant possibilities, and I'm sure if I take just by random 5 people from the audience, they could come up in an hour with 500 ideas how to use genetic engineering to improve classical biowarfare agents, to improve their effectiveness, to ease access, for example, to smallpox, or to create new agents. But all this, I think, is sort of thinking within the box of classical biowarfare, classical infectious agents. And what I want to raise today is the issue that there are many more applications of biomedicine and that may be abused for military purposes. And I just randomly choose 3 projects, one company, one individual researcher, and one research network to highlight where a military abuse might happen today or tomorrow. So the first example is a company, Epicide. Maybe you've heard about it. It's California-based. They make money or want to make money out of producing pharmaceuticals in plants as bioreactors. Pretty straightforward. Last year it was reported that this company introduced a gene for an antibody targeting human sperm cells. So they want to produce this antibody in the plant, isolate it out of the plant, and use it as a contraceptive. I'm not sure whether this will ever work, whether this will become a commercial product. But the mere idea of having a corn, a maize plant, that contains a contraceptive is very tempting for biowarriors. Think about a situation where 2 nations have a long-standing conflict. Or think about a country like former South Africa, apartheid South Africa. They even had a bioweapons program in South Africa 10 years back where they tried to develop a contraceptive that would only target black people to stop their reproduction. I mean, that was at that time science fiction. But with this type of plant, you could produce that, you could breed it into a high-yielding variety, and sell it or even donate it secretly to this country or that population. Pretty easy. Nobody would realize for years until reproduction stops or declines with all the attached social and psychological effects in there. Just to make it clear, I'm sure Epicide has nothing like that in mind. That's a commercial entity that want to make a product. The science is there. It's not science fiction, it's military fiction. I don't know whether anybody now, any military around the world has an interest in it, but there's a very clear potential abuse here. Just one example. The second example is from neurobiology research. I hope nobody here recognizes this guy on there. We came across this name a couple of months ago when we investigated a new chemical weapons program in the US. I will talk more about that in a minute. During that investigation, we found a list of keywords the Pentagon used, list of words using for Medline researchers, just to follow the field of neurobiology, just to follow what kind of new chemical compounds are currently under development that might be used as a chemical weapon to sedate people, to calm people, to put them to sleep whatsoever. And in this long list of 120, 130 search items, there were two names of scientists, and this guy was one of them, because he was working in a field that seems to be of quite of interest for the Pentagon. We contacted him via email, and he was completely taken by surprise and taken aback that his work could have any military application. I mean, this is a poor lab researcher developing a very promising new neuropeptide for anxiety regulation, but he was one of two guys who was so interesting for the Pentagon that they were following him very closely for a new type of non-lethal chemical weapon. I will come back to that. And the third issue, that's the icon, the logo of a research network in Germany on pharmacogenetics. I choose this example because I myself am involved in pharmacogenetics. We are working on at the university on a research program to assess legal, ethical, social implications of pharmacogenetics. Pharmacogenetics is about the genetic differences in individuals, how they react to different drugs. You know, you never react to codeine, but I do, and there are sometimes genetic differences on that. And today there's a hype on pharmacogenetics because it's sort of the first material outcome of the Human Genome Project. And many, many clinical trials around the world are performed to identify specific polymorphisms that are involved in drug reaction and drug action. And during the studies, a lot of ethnic-specific genetic data is generated. And this again comes back to the ethnic weapons. They were already mentioned by Volker Beck. Ethnic weapons are weapons that target one population while leaving another population unaffected. And current wisdom in science holds they are impossible by principle because the genetic difference within a population is much bigger than the genetic difference between two populations. That's what I've heard millions of times during the past 3 years. So geneticists around the world say ethnic weapons are impossible. But during my work on pharmacogenetics, I really dug deep into it, and I found a variety of polymorphisms that are 100% restricted to one population, and you never find them in another population. I can give you examples on TPMT polymorphisms, for example. So there are indeed at least SNPs, single nucleotide polymorphisms, that are restricted to blacks and Asians only, and you will never find them in white Caucasians. I can show them to you. So this current wisdom does not hold any longer. I don't know whether it will ever be possible to translate a SNP into a weapon. So what you would need is a mechanism that this deadly or lethal agent targets especially this SNP. But maybe this is only another 10 or 20 years down the road when that is possible. And then we are really faced with ethnic weapons or the possibility of ethnic weapons. What I want to say here is the hype in pharmacogenetics generates tons of data on ethnic-specific polymorphisms. So the more we do this research, the more data we generate that may be abused in the future for ethnic weapons. I don't know whether any of you already realize that he or she is involved in any one of these projects, but I'm sure we can just flip through the last issue of the EMBL reports, and we will find a variety of projects that maybe one-to-one be abused for military uses. And that's the key point, and that's probably my key mission here today. This is from the editorial from this week's Science magazine by the presidents of the NAS and the British Royal Society. If you read this, it sounds like boring, and we heard that 1,000 times, and it seems to be very clear to everybody. Sure, we have to think about the effects of what we are doing, and everybody would say, yes, I do. But my experience from the past years when I started to work on biological weapons is that especially the biomedical community never ever thinks about what might happen, where is a military potential here and there. Would you have thought about a contraceptive corn that may be abused for military? No. I think it's just not standard in the biomedical community to ask this question, and that's one of the points I want to urge you, that's necessary. Awareness, being aware of what could happen to what you do. Awareness building is probably a key step, and that's what Bruce Alberts and Robert May are asking for. Be aware of what could happen. But while awareness is a key step, I think it's only the first step. And I think scientists must go the second step. Not being aware of what could happen, but to intervene, speak out. It's not enough to be aware that this might be abused, that my neurobiology, for example, could be used by the US Army as a chemical weapon, but be outspoken and say, no, it's a bad idea to do that, because they probably listen more to scientists than to NGO people like me. Okay, that was point one. It lurks everywhere. And point two is about the new desires by Western countries or other countries to get involved in chemical and biological weapons. We all learned at school that biological and chemical weapons are evil. You just don't do that. They are prohibited since 77 years now, and they are just bad. But this old truth seems to change. Now there are good and bad chemical and biological weapons. And I will show you 3 examples of ongoing research. The first one is the already mentioned non-lethal chemical weapons. And this is pretty actual. I mean, 1 week ago, this was used in the Moscow Theater Hall. What we did in the past 18 years is, 18 years, not that long, 18 months, to dig in a US program on calmatives, anesthetic, narcotic agents, convulsants, and long-range delivery devices, so grenades that could deliver gases over 2 or 3 kilometers. They are under development. It's not only research. They are under development in the USA, and that's a clear breach of the Chemical Weapons Convention. It's just prohibited. But because they are deemed to be non-lethal, because there's a need by the Army, and because the biomedical scientists have new and new compounds that might be used, the US obviously just decided to ignore the fact that they are illegal. And that's where I say they start to distinguish between good and bad chemical weapons, and the same applies to oh, I forgot this. That was just one example. One paper we found, there's the full copy, where the Marine Corps University in the US produced a report on different kind of chemicals. And maybe it's not just by accident that the fentanyl that is on the cover, which is basically wrong here, but that was their fault, not mine, that was probably the compound that was used in Moscow last week. So the same is true for biological weapons. There are 2 projects that are going on. One is the development of phytopathogenic fungi to target opium, poppy, or coca plants. So drugs, biological agents to be used in the drug war. This year is Pleospora. That's a fungus that attacks and kills opium poppy. Another one, Fusarium, is under development for coca. They have been developed in the past, now it's about 12 years, by several states, and they are nearly ready to use today. They are also biological weapons. I mean, I've often heard in this discussion, but wait, the war on drugs, isn't that a good cause? I mean, we are all against drug trade. I'm against the drug trade, and I think it's good to do something against it. But I think it's a bad idea to use a biological weapon in the drug war. A good cause is not an excuse to use a biological weapon. I think many of us here agree that the war on terrorism may be a good cause. Why not use anthrax on Al-Qaeda? Because we decided not to use biological weapons under any circumstances. And any war has always a good cause, at least for one of the 2 sides. I mean, it's that simple. Ask one of both sides, and they always have a good cause for the war. So I think it's a bad excuse. And if we start to distinguish here again between good causes and bad causes, who decides that? Between good and bad biological weapons, that's not the point. We can't do that. And the same applies to anti-material agents. The Biological Weapons Convention does not distinguish between agents. Volker Beck made it very clear, the hostile intent is forbidden. Whether you kill people, you kill plants, you kill animals, or whether you destroy material. We have written evidence that also again in the USA, there are some fungi under development to destroy military material. For example, one fungus that was genetically engineered to overexpress polyurethanase that would strip off the special coating of the stealth airplanes so you could see them on the radar and just shoot them down. And that seems to work. I don't know, but it seems to work. They are obviously confident that it will work. There are many things to say about these programs. They are unsafe. They always risk the escalation into an all-out chemical or biological war. But the key point I want to make today is they undermine our current arms control regime. I said it before. We dedicated ourselves not to use, not to develop, not to stockpile, not to produce biological and chemical weapons. And now suddenly some countries start to poke one hole after the other into this treaty. And I think if we start to do that, and if we allow that, and if we contribute as scientists to that, we are on a very slippery slope, which can only end in a new biological and chemical arms race. So I think as scientists, you and we have the responsibility to be outspoken here. So what can we do about all that stuff? I mean, it's ugly enough. The first thing that always comes to mind is a code of conduct, which I think is a good thing. But it's also flawed in a way. I think all the good guys and girls, or how you say that, so everybody who's on the good side would sign on to that and be happy, and the 0.01% of the scientists who decide to do it, they will do it anyway. With a code of conduct, you can't stop a biowarfare project. I think a code of conduct is a very valuable tool to raise awareness, and I think that's what's needed today, to raise awareness in the scientific community, but it does not go beyond. The next step could be a code of conduct for states. And that's a new idea that maybe it's not new, but it was brought forward a couple of weeks ago for the first time by the Federation of American Scientists in the USA. They are now arguing for a code of conduct for biodefense programs, for example. That they have to be transparent, that certain issues should be banned, that you do not work on delivery devices, for example, certain genetic engineering experiments. So a code of conduct for states where the states would sign on to. That would be a bit stronger because this could become sort of politically binding, and it would look suspicious if one country or the other would not sign on to that. I don't know what the German government would say to that, but maybe Volker Beck can later on comment on that. The third point is strengthening the Bioweapons Convention. I won't say a word about that because John Walker will cover that extensively. And the fourth one is a new idea, a legally binding, multilateral, enforceable biosecurity convention. What is biosecurity? It's something that covers all the broad ground between the biosafety protocol on the one side and the Bioweapons Convention on the other side. And there are many things this biosecurity convention could do, and I'm pretty much aware of the fact that I'm here in the lion's den if I talk about the biosafety convention, but still I will go on with this. First thing that could be covered by this biosecurity convention is the national and international transfer of pathogens. Just to make it more difficult to get hold of the most virulent strain of anthrax, for example, which was a couple of years ago pretty easy. It's now a bit more difficult, but there's no international standard and not at all a legally binding convention. That would be one point in here. The second one is just to restrict or at least control or at least make transparent a variety of experiments that would increase the bioweapons potential of some agents. I mean, the simplest thing would be to say, in the moment you introduce a gene that confers pathogenicity, that should be at least notified. maybe it should be prohibited, whatever you decide on. But this broad field should be covered by such a convention. And the third issue is transparency. We must know what's going on internationally in the different labs. This transparency was one of the key points for the protocol to strengthen the Bioweapons Convention, which failed. John Walker will talk extensively about that. That protocol failed, but the transparency could be introduced in such a biosecurity convention. And the last one is research restrictions. Again, it's probably a bad idea to come to the EMBL and talk about research restrictions, but I think it's a valid point. I'm not talking to prohibit pharmacogenomics because in 20 years somebody could make ethnic weapons out of that. No, I'm talking about, are there better ways to do pharmacogenetics without generating ethnic-specific data, without hampering the development of pharmacogenetics? And there are. For one example, pretty simple, is Sequinome now has this method on the market. You can use pooled ethnic diverse samples, 400 samples at a time, and calculate allele frequency in there. So the result you need, you can get without generating ethnic-specific data. And this approach applied to all the other different fields where hostile abuse may lurk. I think that's necessary. And there may be the one point or the other point where we say, this may be such a bad idea, we better don't do that. And that's what I mean with research restrictions. And with this, I end and hope we will have a fruitful discussion on that. Thank you very much.
Laurie?
I could ask a million questions at this point, but I have two that I take very seriously. I hope the answers will be very serious as well. First, to Jan, you listed many things that you thought we might have to control, but in my investigations with biopreparat and with the specific organisms you cited that they had made interesting improvements upon, what they obtained from Western scientists, and in particular, in one case from German scientists, were specific plasmids, specific gene sequences, the kinds of things that no one currently is talking about regulating. How would you go about figuring out a judicious way to regulate the exchange of such things as plasmids or DNA sequences? And if that's impossible, you know, what's your slippery slope? Where do you draw the line? And to John and Volker, the UN Security Council voted yesterday on the Iraq resolution. So UNSCOM, or whatever we're going to call it now, is about to head into Iraq. We found that we were frustrated by a combination of cat and mouse game and the lack of any kind of real detection equipment the last time around. Has anything improved in the 10 years since we last were tromping around Iraq looking for what they might or might not have? Has there actually been any significant improvement in truly available technology that would make the task this time around easier or more accurate?
Jan, would you like to start with?
Yeah, I start with the easier answer. I think it's two things I would like to see. The first one, can you hear me? Is that one here? The first one is within this biosecurity convention, a system of, let's call it laboratory certification. So the transfer of a broad range of items, agents, and even plasmids, that this may be restricted to certified laboratories. That's a system you already have in place, for example, within the US, for transfers within the US, so that nobody, like it happened 10 years or 8 years ago, that a right-wing fascist could just order at the ATCC some plague bacteria. So now it's restricted to certified laboratories. If you could do that on an international level, so you have a certifying body that makes sure that this laboratory is a laboratory at first, and then second could also prove and certify end users, so that any kind of transfer is restricted to these certified laboratories. Everybody can be certified if the laboratory wants to, or they don't do it, but then they don't receive the stuff. So that's one system that pretty much works within one country, and I think that should be workable internationally. The second point is, what I would like to see is a very comprehensive notification system. I'm just talking about notification, huge databases. I mean, it's simple today with the computing power we have on everything. I mean, I've heard 1,000 arguments against it and know them all. But as a principle, to start with a notification system on transfer of everything you need in biology, why not? I mean, you can do it. You can install it at the laboratories, at the suppliers, at the borders. I mean, there are so many data already generated at customs, for example, and export controls and here and there. Put that in one comprehensive notification system. And over time, you can generate patterns. You can suddenly realize that something that happens in that country or in that laboratory does not fit with their explanation they give. There's one precedent to that. One of the first sort of hard evidence UNSCOM found in Iraq was huge amounts of imports of, how do you call this?
Growth media.
Growth media. Sorry, yeah, thank you. Growth media. So, and they couldn't account for that. And they couldn't give a realistic explanation what they did with these tons of growth media. So if you have a good notification system, if you have a good system to track it, and if you have some good algorithms to check back on different countries and laboratories, you might easily come up with some indications that here and there something does not fit the official explanation. So the two answers are: first, it's a certification system on an international level, and second, it's a comprehensive notification system.
Ship a sample to him, he'd have to notify an international control agency that would say yes or no whether that shipment could occur. It would be entered into an international database, or that graduate student's career suffers because he can't do the research that he's going to do in collaboration with Robin Weiss.
Notification is not control. Notification is just you put it in the database. So the first step is... No, these are the two things. The first one is the South African lab and EMBL will probably be certified. The first thing, this regulation or this biosecurity convention comes, EMBL would get a certification. Pretty easy. I mean, every American lab does it today. It's simple, it's easy, you can do it. You do it once and you're certified, and then you get checked upon by the certification body once in a while. But that's standard. And you can do it on an international level. That's point one. And that doesn't refer to any graduate student or PhD student, but the lab certified. The second thing is a notification system. So you do not ask for permission, but you just order it. And this order, which is today in 99.9% cases anyway electronically, is automatically fed into the system. You don't wait for any answer. You don't have to fill in a form, but whatsoever. But it's just entered in the database. And that's it. And this database may be maintained by the same certification body. But so you don't go through a hassle for every single order you do. I mean, I used to work for a couple of years in a laboratory, and I know what's impossible to do, but I also know what's possible to do. But now the second answer is on Iraq, and this is pretty much...
Who of you wants to? John?
Okay. Well, the simple answer to your question, Laurie, is no. On the UNMOVIC, there is no detection equipment. There's no magic box you can wave over a drum or a fermenter that will tell you. So they're not going to be going into country with effective verification equipment, which is going to make their life significantly easier. Taking samples and bringing them back for analysis, well, the laboratory techniques have improved over the last 10 years, but that's going to be some time down the train. They won't have instant results. It's ultimately going to come down to the politics of it, just how cooperative and open is Iraq going to be. When UNSCOM started its work in the early 1990s, and I used to deal with that subject when it started, when we were waiting for the first Iraqi declaration after the passage of Council Resolution 687, the phrase I coined was the dance of the 7,000 veils, waiting for this first declaration. And I suspect we're probably going to see a similar situation once UNMOVIC gets back on the ground.
I think Volker Beck wants to comment.
I want to add one thing. I think it's not a question if your UNMOVIC goes to the Iraq, if they have detection equipment where they can do immediate detection in the Iraq. It's a question to take samples and to bring them somewhere for identification. And you may know that ANSCOM in the second phase of their activities had a laboratory in Baghdad, in the ongoing monitoring and verification center Baghdad, and they had tied up with a lot of laboratories worldwide, which could more detailed identification and detection. And UNMOVIC is going the same way. They plan to also to install a laboratory for screening in Baghdad again, and they already have made contacts to a lot of laboratories in Western and Eastern countries which have special knowledge for identifying agents of concern. If you allow me, let me add one comment also what was said by Jan. You can agree on every type of international convention for transfer and for control, but you have to be sure that any international treaty has to be nationally implemented by legislation. And at the end, I think it's the responsibility of each state to implement a legislation that you really can conduct this method of control and survey. And in principle, export control regulations as exist today for equipment and for agents already doing things like this, as they only have a negative control. And the negative control in the today export control methods are that you only report denials. So it means if a country X denies delivery of equipment or an agent to country Y, it reports this back to the like-minded countries in the control system, so that the delivery cannot be, or the refusal of the delivery cannot be undercut by another country in the community. So you have mechanisms like this, and you have national legislation where you implement what you are allowed to transfer or not, and you have mechanisms with end-user certificates. You have the obligation for the provider of equipment or agents to check if the buyer or the agency that is looking for the equipment or the agent has really legitimate use for this. So you have a type of this mechanism. If they are the best ones, and as they are now only restricted to like-minded countries, if there is a better way to do this, that has to be debated. But I tell you clearly, and you may have heard this from the comments that were given more or less indirectly by John Walker. Presently, the international community, and especially a big partner in the international community, has not big attempts to improve multilateral regimes.
Berthold?
Yeah, the question to Jan van Aken. For me, your definition of bioweapons seemed to be a bit broad, because how would you draw the line? Is it important if something is used inside the country? For example, if the Colombian government would decide to eradicate their coca plantations, is that a bioweapon? And then what's the difference to herbicides or to other things that are currently used? And the second thing is more a note on the problem of possible ethnic weapons. I had the feeling reading an article in Genome Biology, October issue, that there seems to be some political correctness-fueled misconceptions about genetic diversity and ethnic groups. I'm not a human geneticist, but it looks like, or to me, from evolutionary point of view, it makes sense that groups that have been together for a long time geographically also might be genetically more diverse from groups that are further away. So I think this ethnic problem is surely not out of the world, and there seems to be scientific debate about this. And your comment on the SNPs, there's recently the technology of RNA interference, and this is a sequence-directed technology to knock out genes specifically. So I'm not sure. I think we might be not so far.
Yes, on the definition of what a biological weapon is. In the convention, it's pretty clear. There's no restriction to war between countries. There's not even an exemption for law enforcement. There's no exemption at all. Any hostile use. is covered. In Colombia, the situation is pretty straightforward. There's a civil war in Colombia, and one part of the country is controlled by one side, and the other part of the country is controlled by the other side. And the intent was to use it from one side on the other side. So that's a very typical civil war situation. It would be similar to the Russians using anthrax now in Chechnya. I think the viewpoint on Russia and Chechnya is the same like the current Colombian government has on the regions in Colombia. Yes, no, but first of all, I mean, the one point is there a difference between countries or within a country? And the second point is there a difference between plants and humans? But the first point is now addressing just civil wars, wars between countries, or even law enforcement. And I say if the strict reading of the convention tells you even law enforcement would be prohibited. The police cannot come on my field, if I'm a farmer, on my maize field, and use fungi on them. They could destroy it, they can uproot it. That's not prohibited. They can use herbicides because there's an exemption for law enforcement in the Chemical Weapons Convention. And the Chemical Weapons Convention does not cover use against plants. But in the Bioweapons Convention, both items are covered, use against plants and law enforcement as well as civil war. I think that's one of the arguments that has been made. So the Clinton administration said, if the government agrees to use it, then we're going to use it. But they changed their mind. In the year 2000, the Clinton administration backed out of that. And with a reference to the Bioweapons Convention, they said, first, stop the program in Colombia. And for good reason, because if you start allowing it to be used within a civil war, I mean, then you're dead. There are so many wars out there which are seen by one side as a civil war. Look at Russia and Chechnya. I think that's pretty straightforward.
I think Volker Beck wants to.
Yeah, if you allow me, in this point, I disagree a little bit with what was said by Jan van Aken. And you have to be very cautious because the Biological Weapons Convention is not talking in law enforcement. There's terminology from the Chemical Weapons Convention, and the Biological Weapons Convention is talking on peaceful uses. Now, if in this special case of the destruction of drug crops, I think you have to be very cautious interpreting this as a biological weapon, even in an area where an internal struggle between different groups, government on the one hand and other groups is going on. I think it would be more critical if you use agent for eradication of a crop which really is used for human consumers or for animal consumers in this area, because then you are going to the basics of the populations living there. But if you're destroying a drug crop, which we know exactly that we have in the United Nations a lot of agreement and of resolutions that this is a global effort to destroy drug growth and illicit drug traffic. So I think that's very difficult, I think, in this case to argue that this is the use of a biological weapon, because it's nothing where people live from by daily eating or animal live from daily eating what you destroy there. If you go on the crop side, and then I can debate this differently, because there's another approach to this, but not if it's going on the destruction of drugs. And in this case, independent from this, we have a similar case for poppies, which is going in the Asian region, where there's agreement with the governments, and there's no discussion about this. You can otherwise use the same arguments there. So it's a difficult issue.
The coca plant certainly helps you when you're hiking at 4,500 meters. But anyway, Ronald?
I have a question for Dr. Beck, because your talk has made me very pessimistic about the outcome of the Iraq inspection, because at the core of your talk is your definition of what is a biological weapon. And if I check my notes, probably the same reason that you find that the definition has been so stable since 1925 is also the reason that we're going to run into a totally predictable problem, is because at the core of it is whether the type and the quantity of the equipment can be justified for peaceful applications. And you've given numerous examples of aerosol generators and DNA synthesizers and all sorts of equipment which obviously can be justified for peaceful application. And doesn't that mean that it is entirely 100% predictable what's going to happen? No equipment will be found which can not also be found in the city of Heidelberg. And then the next issue is, what is the application, or what's the intention with that machine? But there's no way that inspectors can evaluate the intention by just looking at laboratories, right? And then you're back to politics, and what's the intention of Saddam Hussein?
Okay, I think from reading this it was not so clear, so I have to repeat it. What is prohibited is to possess or to store or to retain microbiological agents. That means bacteria, fungi, viruses, or toxins in types and quantities that have no justification for prophylactic, protective, or peaceful uses. The equipment issue in the definition of the weapon is not related to technical equipment to produce these agents. The equipment part of the definition is related to equipment which you need to have for the dissemination of these agents. That means either a real weapon where you fill it in, or an aerosol generator, or a spray tank, or something like this. So the possession of equipment which you in laboratory use or in production use is not prohibited by the convention.
But how about the aerosol generator?
With the aerosol generator, that is an interesting question, because in the Iraq they have helicopters with aerosol spraying devices. Because in this country you find a lot of date plantations, and they spray insecticides on the dates. And they have, if you look for the agriculture, they have a lot of monocultures where you need to spray herbicides. So it's a question if the aerosol generators which you have connected to the helicopters are really usable for producing particles in this size area of 1 to 5 micrometers. That is the inhalable size area. And that's going only for human infection, not for crop, that's different. But in the Iraq, what was found there was aerosol generators which were constructed in a way that you can only use it for agricultural purposes. So if it's very clear that you can only use it for these purposes, then it's not against the convention. And it's not even not a dual-use equipment in the sense of how it's defined in the convention. So that is one of the crucial issues. I think if in Iraq, if you go to a facility that can produce large types and quantities of vaccines, and somebody says that is a facility, that is a biological weapon facility, you have to prove if they really have produced in large quantities agents for which you can use as biological weapons. Simple issue, if they really have produced what they didn't do, anthrax vaccine in this, so that is legitimate. If they have produced anthrax viable strain which can be used, or spores which can be used as a weapon, it's illegal. So you really have to get hold of the smoking gun. And if you don't do this, you are lost.
So you're also pessimistic.
I would say in a similar way, as I said it just before, UNMOVIC will not be possible to do more than what has ANSCOM achieved. And ANSCOM has achieved, to make this clear, I'm not negative on ANSCOM. ANSCOM has achieved a lot of. It had verified that there was no nuclear program that led to a nuclear weapon. They have uncovered most of the chemical and destroyed, to our understanding, most of the chemicals that were weaponized in the Iraq. There's some small doubt on one type of agent. And they have uncovered that there was a biological weapons program, but they didn't get hold of any weapon containing a biological agent or any agent in quantity that they can prove what really was going on. All what was connected with the biological weapon program is that ANSCOM identified there were facilities where you can do it, there were research done, and there were declaration by the Iraq how much they have done. And there was one thing that was not well explained. for what purpose these huge amounts of gross media, which were found still in the Iraq, that was half of what they had procured, for what purpose it was for. So, but they didn't find the smoking gun.
I think John wants to comment on that as well. Or
Well, just on
as a-
2
yeah.
points, but Volker's last point, in actual fact, they did discover on one occasion a smoking gun. There was a type of bomb, an iron bomb, dropped by aircraft the Iraqis used in their BW program. And those weapons had special linings. And a sample was taken from that, and they found traces of botulinum toxin. So on one occasion, the UNSCOM inspection process identified, if you like, the smoking gun. But as Volker said, hitherto, what they got was a very strong smell of cordite throughout Iraq. And that was the point I wanted to comment on, that the dual use problem is significant. There's no getting away from that fact. So what do inspectors have to do? And that is look at the context of which that equipment and activity is taking place. Now, what we found in the UK when we conducted a large number of trials with industry about how you do inspections in the biological area, we found that the documentation and the explanations provided by the facility personnel to explain their activities was credible scientifically. It made sense commercially, and there was a coherence to the information, and you make a judgment from that this is all plausible, it makes sense. Now, that's most of what you can do. In Iraq, it's a complete absence. Now, in the second UNSCOM inspection, there were 10 facilities inspected, 9 of which the team were able to conclude fairly easily that these probably weren't connected with the BW program. But one of them, the team were pretty certain had to be connected to an offensive BW program, even though at that stage they hadn't found a smoking gun. And what was it that gave the clues? The design of the facility. Why would you have revetted structures in something purporting to be a single-cell protein plant? The person put up as the plant manager knew nothing about single-cell protein, couldn't answer any of the questions. So the complete absence of credibility was one of the key factors that said there is something wrong here. They have not answered the questions. And that's what the inspection process is designed to do. If the Iraqis are transparent and open, then the problem goes away. But if they persist in being evasive, tell outrageous lies, then we're back into political problems.
If you allow me one sentence, that is one of the additional benefits. As long as UNSCOM was in the Iraq and had this ongoing monitoring center in Baghdad, we knew which were the facilities of concern, and we had every time access to the facilities of concern. And with the disappearance of UNSCOM from the Iraq, we lost the track of what's really going on.
Hi, my name is Jennifer Michelle, and I just wanted to say that I think there's kind of a narrow focus happening here, and I think it's realistic because I think we're talking about molecular biology, which is a very precise science. But when I started hearing people talking about Colombia and saying it's a civil war, despite the huge amount of international funding coming from my government and how that's been linked to huge paramilitary abuses, when I start seeing how there have been decades of spraying in that entire extremely diverse part of the rainforest, and it's been only herbicides and only things that have been okayed, and yet the devastation it's causing to the indigenous farmers has been well documented, and it just moves from place to place. When you talk about how it's only going to be a toxin that's an herbicide, and the way there are tip-offs going on there that have also been well documented, so that the major plantations for the coca are completely avoided because they're tipped off because of the links that nobody's really discussing. So it lands directly on the small farmers who have nothing else to plant, and so they are dying and they cannot feed their children. Then I think the naivete level here is really profoundly seen. This is not just about herbicides. This is not something that has not been untried. It's been tried and proven for decades. I mean, talk to Larry Thompson at Refugees International, talk to Hiram Ruiz at U.S. Committee for Refugees, talk to anybody in the Latin America Caribbean section of Human Rights Watch. This is not a scientific issue. This is a human rights issue, and it is very well documented. The other issue I want to bring up has to do with my incredible bias against my own government at the moment, which is, while I would like to believe that I think Iraq is the major evil on the planet, and while I would like to believe that Al-Qaeda is, I do not. And when I hear Jan up there telling me how the U.S. government has already violated one treaty, when I talk and hear people talking about the securities resolutions at the UN, and I see you're all missing the little dance that you just were led through, in my opinion, by your noses by my lovely government, through the UN, my government did not want to negotiate with the Allies. My government did not want to use the UN whatsoever. And the minute my government, and the little troll that is now running it, decides that you are expendable and immaterial for negotiating, the Allies and these lovely conventions that you have set up are going to be out the window. And I think to hear you talking about this as if my government is on your side and as if my government is sitting there as one of the good guys, I think it's a naivete beyond belief. And maybe I'm paranoid, and maybe I've watched way too many X-Files, but I am sorry. Do not trust them. And to sit there thinking that they will follow your negotiations and your treaties as if they give a goddamn about your social justice issues and about any of it is the most profoundly frightening naivete I have seen. And I do not know where you have been in the last century, and I do not know where you have been in the last decade.
Thanks for the comment. I was almost making a remark at the beginning about the American government, which I sort of didn't, because we actually wanted to have an American on the panel as well, but they were all very busy these days for obvious reasons. So I think John wants to comment.
It's been one of the great burdens of my professional career working in the Foreign Office to spend more hours than I care to remember in meetings with US officials, arguing about the scope and nature of efforts to strengthen the BWC. Now, you would think with the good old Western allies, we were spending more time worrying about and talking and negotiating with, say, the Russians or whoever. That's not the case. The whole reason the protocol failed in the end was because the United States determined that it wasn't in their national security interest to do so. But there were a large number of other countries who were hiding behind the US position who were actually quite happy that this thing fell apart. So we are not naive. We have to live in the real world and spend time trying to persuade countries, including the US, to see things differently. And that is not an easy task. And ultimately, as we found with the protocol, all our efforts failed. The UK, speaking really with official hat on here, we spent a lot of time and effort trying to find ways in which the US could be kept on board. Ultimately, we failed because the US decided to do, as you say, its own thing. So I don't think we're naive. We're actually brutally aware of realities.
Okay. To add to this, you know, the history tells that I think we are in a state of development with the present, and when I say present, I don't mean the Bush administration, so the development of US politics on this issue in the last decade. We are in a phase where I think the tendency in US politics are a little bit diverting from how European countries or other countries are looking on global security. But that cannot mean that we move out of the process because we believe that there are partners in the process that are doing wrong. Our approach is that we need to have a platform to talk to all, and also to include in the context of the states parties conventions, the states where we have suspicion that they are breaking the treaty. Because we need to have a platform to discuss with them internally this issue and to convince them what's going wrong. And for this, you have to accept that you have to deal with partners where maybe a disagreement for some period, but we know exactly that periods in politics may be a decade or so. But that cannot mean that we can cut off discussions because we never know how far and how quick positions change. And so it's a common interest, and even the common interest of your government, to keep this process going on. But in this case, it was very crucial because the United States is the biggest contributor to biosciences and life sciences, as well in academia, as well in biodefense, as well in industry. And if we wouldn't have kept the United States in this process, exactly the countries of concerns would have moved all out, hiding behind the United States. And for this reason, we are not naive. I think we are a little bit more pragmatic as we really sometimes try to tolerate ourselves.
Maybe I also add something here. John and Volker, I think you're deadly wrong. And I think it doesn't help you to engage in anti-Americanism. You don't do that, but many people do. I think it doesn't help. The bad guys today are the British and the German government, because there is a way out of it. There's a way to go ahead without the US, and I'm arguing that for 2 years now, and I know you are on a completely different track, and I think you're on the wrong track. The US never signed the Geneva Protocol for 50 years, and still the Geneva Protocol was a very, very powerful instrument. They did not sign anything in the past years, and it was right to go on with Kyoto, and it was right to go on with the Landmine Treaty. These are helpful treaties. So I think also in this regard to the biological weapons, go ahead without the Americans. They will come on board later. When I talk about the Biosecurity Convention, I'm talking about a Biosecurity Convention without the US in the beginning. Start the process. I always say, get some like-minded countries, start a new process with like-minded countries, and after a while the Americans will come on board, and if they don't come on board, they will still, like with the landmines, like with the Kyoto, they will do their homework. I know that you completely disagree, but I think you have to do something here, and you can't just be taken hostage by the US government today. It's wrong.
Does anybody want to comment straight on this, or shall we yeah, please.
This is for you, John Walker. Your presentation today about the importance of the treaty for food security and for public health. Surely, Laurie Garrett was telling us yesterday about the role of public health problems and disease in global security, and the fact that the Americans are becoming very, very aware of that situation. Are you optimistic that perhaps the change in American thinking about global food and health security is going to inform them in decisions like whether they choose to sign the Bioweapons Treaty or not?
The simple answer to that question is no, because as we've just been discussing, the attitude of the current administration to multilateral solutions to international problems, well, I think the sympathies of the room are well known at this point, and at a personal level we've got a real problem there. On the Biological Weapons Convention side, there were, for many years, even before Bush came along, there was this animus almost against BW arms control. We had to spend a lot of time persuading, cajoling, arguing why it was important to engage at an international level to strengthen the treaty through international efforts. We get everybody to sign up on to. Now, there are lots of frustrations in that process, as are evident by the fact that 10 years we've got nothing to show for it. On these other areas of international security, the concept of what constitutes international security, of course, is broadening, and maybe over time that might percolate through where people do realize that they have to work, the US administration do have to work with the rest of the world to deal with these problems. But I'm not sure that people like Paul Wolfitz or John Bolton really see that view. So I think the short answer is, yeah, I'm pessimistic.
You really think that the last 10 years didn't bring anything, or do you think that maybe a new US administration would actually realize that there was 10 years of a big effort and that might make a joining and possible?
Well, I think if we again look at the history, until 1992, the official US administration position was that we know of no way of making this treaty verifiable. The US were prepared to accept the verification expert process that ran from 92 to 94 precisely because it didn't have a negotiating mandate. It was a scientific technical review to evaluate measures. What were the good ones, what were the bad ones? The only reason that we got, one of the principal reasons we got a mandate to go off and negotiate a protocol was because we had a Clinton administration, which was ostensibly very enthused about the prospect of doing something. So even though we had an administration which at public levels was committed to a protocol, 8 years they had. And a lot of the problems we faced were precisely the detail of how we were going to make this thing work, arguing with the US officials. And so when we had the Bush administration come along, it was almost certain what was going to be the outcome, though there was always this tantalizing prospect that maybe just perhaps, maybe in the blue moon they might just sign up to it, because there were certain indicators that they might have been prepared to do that. So it kept us all clinging on. So there's been a long-standing animus against that. So I think even if we get a new democratic administration, the chances that the US are going to say, now where's that protocol, where do I sign it, I think is absolute zero. Because the institutional objections and difficulties with the whole concept that exists within the Department of Defense and also in industry, represented by the corporate level pharma, are such that it's very difficult to see how we're going to make progress. Now, Jan's quite right. The US took 50 years to come on board the Geneva Protocol. So someone who's trained as a historian, well, yes, we can wait. Things will change. it's actually ahistorical to say things will never change because they do. Just it's very slow. I'm not sure where that leaves me, but
well, it's probably better be a historian than a geologist, because on a geological timescale, that would be. I think Giuseppe had a question.
I had a question for Jan van Hagen. So coming back to your point about ethnic weapons, right? So pharmacogenomics is only one of the applications of the mass large-scale genome profiling by SNPs. And there are situations in which ethnic groups which have been living in partial isolation from the rest of the community, it's actually very useful and very important to genome profile them with SNPs to get a clear, because this could help for a variety of association studies for complex multi-gene diseases. So in the view of the potential benefit of this research, do you really think it's realistic to think that these programs on which we have almost already embarked will be stopped because of the fear of ethnic weapons?
No, again, I thought I said it hopefully twice, and now I do it again 3 times. It's not about stopping all these programs. Then you could close down EMBL. That's not the point here. It's not about stopping pharmacogenomics, and it's not about stopping generating ethnic genetic data. It's a way of doing, a way of achieving.
For pharmacogenomics, that there would be ways to do it, which of course would not involve the ethnic side, which is great for pharmacogenomics. But there are applications in which you may have to resort exactly to the ethnic information if you want the study to be useful at all.
Yes.
And those, of course, are critical points.
Again, but then you have to look how dangerous is it in terms of biological weapons. And what I know from these kind of ethnic studies, Tristan da Cunha and what's out there, I don't see so much of military abuse. I mean, maybe there's a country that has a problem with Tristan da Cunha and may be interested in that specific ethnic data.
you better not. It's a British dependent territory.
Sorry. Okay, so but from what I know, and I only know several examples, I don't think these kind of studies pose a risk in terms of military abuse. That could change over time if more and more ethnicities are under scientific scrutiny, but my understanding is that this concept is already outdated, and for example the Estland project, I figure it will never start because the scientific validity of this project is questioned by the scientific community itself. Even the Iceland project is coming under criticism because what the pharma industry needs, and now I'm coming again back to pharmacogenetics, is extremely good phenotyped genotypes. And the extremely good phenotyping, you don't get it in an overall population like Iceland or Estland. You get it from very specific approaches to get a small part of a given population. And that could even be an ethnically mixed population that is phenotyped under very certain quality standard criteria. So I think this concept may be outdated already, but you're absolutely right. There will be many cases where you say there's a very clear benefit and there's a very clear military abuse, and we do not have an alternative. But the first step is think about it, talk about it, be outspoken about it, and see whether you can't find an alternative. And we don't have the awareness yet. We don't have people being outspoken against it, and nobody's looking for alternatives. So if we are there, we are already 3 steps further, and then we can come back to the question, I think.
If I remember right, there was Rob Ridley who had it, or that's been done. Then there was a question right here. It's been done as well. Okay, so please.
Hi, I'm also a historian. So I'm totally baffled by this idea of ethnic weapons, because it would make sense to me if peoples lived together on certain spaces for prolonged periods of time without mixing, which they don't ever. I mean, there are some very small communities like Iceland, which are the exceptions that have had very little interchange for centuries, but there are very few, and I can't think of any group that would be large enough or important enough to be targeted. That, you know, as far as I know, you don't find a black person in America that hasn't had some white ancestors down the line. I mean, the South African government spent half a century trying to find some decent classification of races, and never really did. So, how should this work?
Could you just wait to be past the microphone?
You want to answer on that?
Let's take one example, that the majority of black Africans are homozygous negative for Duffy antigen, which is a chemokine receptor on red cells. They still express it on certain endothelia and in the kidney epithelium. There are a lot of small molecular weight drugs currently being designed to interact with chemokine receptors for cancer, for cardiovascular disease, and as anti-HIV agents. Now, if you can devise antagonists, you devise agonists, and it's not beyond the realms of imagination to devise something that would act rather as Duff is believed to act, which is a sink for chemokines. And black Africans would not have that sink, and therefore there might be 100-fold difference in toxic dose between a black African and the rest of the world. Now, most American blacks will be a mixture of homozygotes and heterozygotes for this particular thing, because their genes there. I mean, my doubts is that we know an awful lot about ethnic genotyping. I mean, we've just completed a paper, it's not quite sent off yet, on the evolution of human herpes virus 8 in different communities. And we particularly looked at its diversification over the last 2,000 years in Ashkenazi Jews. Now, I'm an Ashkenazi Jew, and we could show that this virus, which goes in families, transmits from mother to child and not through father to child, which you wouldn't really expect, because it diversifies according to the SNPs in mitochondrial DNA and bears no relation to the Jewish specific sequences in Y chromosome DNA. I mean, we're doing all this. We know a lot about, there is a lot of what's called genetic anthropology out there. And so the genie's out of the bottle. Maybe I should sign up to some agency to get some research support. So don't worry, I won't.
But to come back to your question, in a way you're right. I mean, you probably refer to the fact that there are no clear borders between ethnic groups, and that's pretty obvious and pretty clear. But if you talk about a weapon, you're not talking about killing 100% of one group and have 100% survivors in the other group. It's probably enough to target 5%, 10% of the other group, if you're only sure that nearly 0% of your own population is not affected. And you can find lots of that. Even if you say, even many blacks in former apartheid South Africa would have the one or the other white gene, there would have been enough, a very high percentage, which did not. So it's not necessary to win a war to kill 100% of the enemies. 5% would be devastating already. So we are talking not about 0 to 100%, we are talking about 0 to 5%, 0 to 15%, something like that. And there are always some gray areas, and maybe one or the other from your own army will fall ill, but that's collateral damage.
Well, I think on that note we can finish with a discussion that I would have brought up if we would have more time, because it seems to be a discrepancy between the call for transparency from some people and obviously the call for secrecy from some other people.